In The Codebreaker, Walter Isaacson takes you on a thrilling journey into the heart of a scientific revolution. Have you ever marveled at the intricate code of life, DNA, and wondered if we could ever truly understand, let alone rewrite, its script to combat devastating diseases or even shape our future? This is no longer the realm of science fiction. The book chronicles the remarkable story of Jennifer Doudna and the discovery of CRISPR, a groundbreaking gene editing technology that has given humanity an unprecedented power, the ability to alter the genetic makeup of ourselves and potentially all future generations. Isaacson masterfully shows that this new power, while holding the risk of unintended consequences, offers immense benefits if navigated wisely. The Codebreaker illuminates how this technology emerged, the brilliant minds behind it, and the profound ethical questions it forces us all to confront, ultimately transforming your understanding of life itself and your role in shaping our genetic future. The journey to one of science's most significant breakthroughs began not in a high-tech lab, but with a young Jennifer Doudna's insatiable curiosity about the natural world in Hawaii. Intrigued by everyday mysteries like why sleeping grass curled when touched, her fascination was further ignited by James Watson's book, The Double Helix. This book didn't just explain the discovery of DNA, it revealed that science was a human adventure, full of detective work, competition, and collaboration. This early inspiration set Doudna on a path less traveled, focusing on RNA, DNA's often overlooked molecular cousin, at a time when most of the scientific world was fixated on DNA. Her persistence in studying the structure and function of RNA, driven by pure curiosity rather than a specific application, laid the crucial groundwork for her later, world-changing discovery. This highlights a core theme of the book: fundamental curiosity-driven research, often exploring obscure corners of biology, is the seedbed from which revolutionary innovations like CRISPR can unexpectedly bloom. You learn that breakthroughs are often born from a persistent desire to understand the why behind nature's wonders. Building on decades of genetic understanding, from Darwin and Mendel to the unraveling of DNA, science eventually turned its attention to the intricate workings of bacteria. It was here, in these ancient microbes, that researchers like Francisco Mojica, and later, scientists improving yogurt cultures, stumbled upon strange, repeating DNA sequences. These were CRISPRs, or clustered regularly interspaced short palindromic repeats, a sophisticated adaptive immune system that bacteria use to fight off attacking viruses. The real magic happened when Jennifer Doudna, collaborating with Emmanuel Charpentier, meticulously unraveled how this system worked. They discovered that a CRISPR-associated enzyme, Cas9, acts like molecular scissors, guided by a piece of RNA, called CRISPR RNA or cRNA, which is processed with the help of another RNA called tracRNA to a precise spot in a virus's DNA to chop it up. Their masterstroke was engineering these two RNA guides into a single guide RNA, or sgRNA. This invention transformed CRISPR Cas9 from a bacterial defense mechanism into a programmable tool. Now, scientists could easily tell the Cas9 scissors exactly where to cut in any DNA sequence, simply by designing a matching guide RNA. This made gene editing remarkably precise, efficient, and far more accessible than ever before, opening the door to rewriting the code of life. The development of CRISPR Cas9 almost immediately ignited a beacon of hope for millions suffering from genetic diseases. You see, this tool can be used for what's called somatic cell editing, where doctors can correct faulty genes in a patient's specific body cells, like blood or eye cells, without affecting their reproductive cells, meaning the changes are not passed to future generations. The book details early pioneering efforts in clinical trials showing remarkable promise for conditions like sickle cell anemia, where Victoria Gray became the first person in the U.S. treated with CRISPR, experiencing a dramatic improvement in her health. Similar advances are being made in fighting certain cancers by editing a patient's immune cells to better target tumors, and in treating some forms of congenital blindness by directly editing cells in the eye. This power to precisely correct genetic errors offers you and your loved ones the tangible hope of conquering diseases that were once untreatable. But as Doudna and Isaacson explore, this healing promise also ushers in more complex questions, when considering changes that could be inherited. The true ethical crucible for CRISPR technology lies in human germline editing, which involves making changes to the DNA of sperm, eggs, or early embryos. Such edits would be present in every cell of the resulting individual and, crucially, would be passed down to all future generations, permanently altering the human gene pool. The book recounts the shockwave sent through the scientific community when Chinese scientist He Jiankui announced in 2018 that he had created the world's first CRISPR-edited babies, claiming to have made them resistant to HIV. This event, widely condemned as premature and ethically fraught, catapulted the abstract debates into urgent reality. You're confronted with profound dilemmas. Where do we draw the line between treating devastating diseases and enhancing human traits like intelligence or athleticism? Who gets to decide what constitutes an improvement, and what are the risks of a genetic supermarket, where only the wealthy can afford enhancements, exacerbating social inequalities? Doudna herself, deeply troubled by these prospects, even having a nightmare about Hitler wanting the technology, became a leading voice in calling for caution and public discussion, spearheading meetings akin to the historic Asilomar Conference to grapple with these issues before the science outpaced our ethical frameworks. The Codebreaker vividly portrays science not as a sterile laboratory process, but as a deeply human endeavor, pulsating with intense competition, brilliant collaboration, and high-stakes ambition. You witness the fierce race between Doudna's group, Feng Zhang's lab at the Broad Institute, and George Church at Harvard to harness CRISPR for editing human genes, leading to patent battles and the formation of rival biotech companies like Editas, Intellia, and CRISPR Therapeutics. This competitive landscape, while sometimes fraught, also spurred rapid innovation. Then, the COVID-19 pandemic erupted, and this very same scientific ecosystem demonstrated its remarkable capacity for crisis response. Doudna, Zhang, and their teams quickly pivoted, repurposing CRISPR technology to develop rapid diagnostic tests like Mammoth's Detector and the Broad's Sherlock. They also began exploring CRISPR-based antiviral treatments. This crisis revealed how scientific rivalry can coexist with a shared commitment to the common good, leading to unprecedented openness in sharing data and accelerating solutions when humanity faces a collective threat. The main takeaway of this book is that Jennifer Doudna's co-discovery of CRISPR has catapulted humanity into an age of unprecedented biological mastery, arming us with a powerful tool to potentially cure diseases and reshape our genetic destiny, but also demanding profound wisdom and collective ethical deliberation. Walter Isaacson's The Codebreaker not only illuminates the elegant science and the human drama behind this revolution, but also empowers you to understand the immense promise and the complex perils of gene editing. By grasping the journey from basic research to this transformative technology, you are better equipped to participate in the crucial conversations about how we, as a species, will choose to wield this code-breaking power for the future of the human race. All right, that's a wrap on today's summary. Thanks for listening. If this book summary sparked your curiosity or gave you new insights, don't forget to like, subscribe, and hit the bell so you won't miss our next summary. Keep reading, keep growing, and see you in the next one.